before I start, I do want to thank the foundation. They've been hugely helpful. The phase 2 data that I'll talk briefly about, they help support that clinical trial with funding. They help encourage people to be involved with that. They continue by a broad range of efforts to give us tools, to give us access to, improve means to reach potential patients in the study. And so, your support of them has been hugely helpful in our case specifically, but I believe for almost everybody here on the panel to be able to be positioned to run the trials that they need to support approval. We're a publicly traded company. You may have heard me talk as Essentialis over the last couple of years. Soleno was formed by the merger of Essentialis with a publicly traded company. I have to put up these certain notices and disclaimers, so there you go. And I have to be careful in what I can say because we can only talk about things that we've made public to not only to all investors, and so we got to be careful answering even questions I may not be able to answer. But Diazoxide, which is mentioned here, is a historically approved product that was approved to treat some very rare diseases in adults and in young children that are hypoglycemic conditions. So these are conditions where the patient experiences recurrent very low blood sugar. And the drug is used as an oral suspension to treat those patients, and they're dosed either 2 or 3 times a day to treat those conditions. And they occur about 1 per million individuals in the U.S. or elsewhere in the world as well. But there is experience and safety data that helps us in making decisions about our drug. Diazoxide choline controlled release tablet, or DCCR, is a once-a-day tablet formulation of a salt of that. We've characterized it in a series of 8 clinical trials. So you heard about phase 1 trials. We ran about 5 of those. Heard about phase 2 trials. We've run several of those as well. And those studies were done in obese and in dyslipidemic, or patients with lipid disorders, and in PWS patients. I'll talk about that data in just a minute. And we've treated more than 200 patients across those studies, and we've got it protected by multiple issued patents. When you think about appetite and hyperphagia, appetite in its most basic sense is regulated by a couple of sets of neurons in the hypothalamus. It's a very small region of the brain. And essentially what you see are the 2 sets, one set of neurons called the NPY-AGRP neurons, which are essentially the accelerator on appetite. And then the POMC neurons, which are the brakes on appetite. And they're regulated, for the most part, by leptin and insulin. So these are hormones produced elsewhere in the body, leptin by fat tissue and insulin by the pancreas, to regulate these. And insulin's supposed to regulate it so that, after a meal, your hunger level drops, and because insulin rose. And then when insulin drops longer after a meal, your appetite can begin to build again towards the next meal. And leptin is sort of the longer-term picture of how much fat stores you have. So if you've eaten well for a long period of time, your appetite is supposed to be suppressed somewhat by this. In Prader-Willi syndrome, because of the loss of at least one of the genes in that Prader-Willi critical region, essentially the situation that you see is that the accelerator's down and the brakes aren't working so well. And that's really partly because there's probably some, both of these 2 molecules, NPY and AGRP, that are made by the accelerator. One's the absolute strongest appetite stimulant that's been characterized, and the other kind of disrupts the brake system. So what happens then with our drug is we mimic or amplify the effects of leptin and insulin, these 2 hormones that regulate these. We amplify those effects to take the foot off the accelerator somewhat and to put the foot on the brakes a little bit more and to reduce hyperphagia by that means. So that's essentially how the drug works. And it works, very basically at the level at which these neurons, the accelerate and brake systems are regulated. So we ran a study that I've talked about before, but I'll talk about it briefly again. In the study that we conducted in Prader-Willi patients, we ran this at the University of California, Irvine, which was close to where we were or are. And essentially what happened is we had 10 weeks in which patients knew they were on drug. We were raising the dose of the drug and then treating them stably for the remainder of that period. And then in the second phase, here in the double-blind phase, half the patients were switched to placebo and half stayed on active drug, and nobody knew. The investigator didn't know, the patient didn't know, the family didn't know. We didn't know whether they were on placebo or whether they are on active. And then we looked at what happened in that period. And then some of them came back for a 6-month treatment period. This looks like it's immediately following, but there was a lag there in which they were off a drug and then came back on and were treated for 6 months longer. So when we've talked about how do we measure hyperphagia, we use a questionnaire that asks a series of questions to the parent or caregiver about about the child's food-related behaviors. We're not measuring how many calories they're actually consuming. And what we saw is as patients began to dose up on our drug, we saw a significant improvement in hyperphagia, so that at the end it was between 30 and 35% reduction in hyperphagia. It was highly significant. And not all the patients were at the same dose, so at the highest dose, the dose that we intend to target on average in our future trial, they were even a more marked improvement in that. And then what we saw is when the patients got switched over to placebo, they began to move back towards where they were at baseline, and those who stayed on DCCR tended to have their improvement in hyperphagia be maintained. We looked at body composition, and we saw what's in green here is the whole population of patients who finished that open label phase, or the 10-week phase. We saw that their body fat was reduced about almost 4%. Their lean body mass increased a little more than 5%, and that ratio of lean body mass to body fat went up almost 10%. At the dose that we're targeting in the future study, they had about a 6% loss of body fat over 10 weeks. They gained about almost 9% of lean body mass, and that ratio improved by almost 17%. So all of these were highly significant, and we saw the same things occur when they were on it for 6 months. So it was a reproducible effect. One of the notable things that we saw in our study, and mechanistically we know why this occurs, so it's not surprising when you understand the mechanism, unrelated to the hyperphagia effect. At baseline, we looked at a series of behaviors, but in particular, we looked at aggressive behaviors in these patients, and about 70% of the patients at the beginning of the study had aggressive behaviors, and by 10 weeks of treatment, only 30% of them were displaying those behaviors any longer, and they had to completely stop the behavior before we noted it as an improvement. They couldn't, they're doing it less often. There's only one time they're throwing objects during the week as opposed to every second day. Nothing like that worked. They had to really completely stop before we showed an improvement. And so again, this is highly significant. And we think in the long term, if this is reproducible, this is a major benefit of the drug on top of the hyperphagia. We looked at lipids, so this is triglycerides, total cholesterol, the good cholesterol, HDL cholesterol, LDL, bad cholesterol, and then non-HDL, which is sort of all of the fractions of cholesterol that contribute to clogging of arteries, if you will. And we saw improvements in these, and these were again expected. We know exactly why this occurs, and it's expected. So if they stayed on through the double-blind phase, there was about a 45% reduction in triglycerides, which is positive, reductions in total cholesterol, improvements in good cholesterol, and so on. we understand the safety profile of our drug, partly because we've run 8 trials now, but also because the parent molecule has been used for some time, and there's probably 120,000 patient years of data, and most of that at doses that are above the doses that we'll use in Prader-Willi. So we understand the safety profile quite well. In the development of our drug, we haven't seen any new safety issues over and above what was understood from the historic use. I already said that the doses are at the low end or below the labeled range for that product that we use with our drug. And we've seen a range of benefits: the hyperphagia improvement, the improvement in aggressive behaviors, reductions in increase in lean body mass and reductions in body fat, and improvements in cardiovascular risk factors. I'm not going to go through the kind of detail that the prior speaker did about the design of the trial, because we haven't made that much of it public, but we are, as Teresa noted, we're ready. We're in the later stage planning phase for a phase 3 study of DCCR in Prader-Willi patients. There'll be multiple sites across the United States. We're in the process of working with those sites to get contracts in place and so on. Primary endpoint will be a hyperphagia improvement from their baseline measurement. It'll be 3 months of double-blind treatment in this study. We expect to start by the end of 2017, so we will, stay tuned. We will be, when we can make it public, we will be making it public, and we'll have multiple means by which we try to communicate with you. Concerning the trial, and on clinicaltrials.gov, it'll be registered there with more detail there about it, including the sites. So with that, I'll stop.
Any questions for Neil, or you want to think about it and ask at the end?
Yes.
In a perfect world, what would be the ETA for completion?
I'm trying to think whether we've made that public or not. I think we haven't made it public, and much as I'd love to tell you, I can't. But it's a nearer term than I probably would have been saying even a year ago. So that's good, but we have to get this trial done. And following this, there's a open label treatment trial that patients roll from this one into. And again, we'll have finished that before we can put together that NDA that Teresa mentioned. So.
Oh, one more question.
Yes.
Did you say what age range did the phase 2 and what age?
Phase 2 went down to age 10. And I don't think we've made public the age range down to which we can go. Partly that there's still a little bit of negotiation going on with FDA, but last year or the years before, I was a private company. I could tell you what I thought right away and not have to sit there and ask myself the question, what's been known? So, yeah. Yes, Lauren.
You mentioned that you understand maybe what might have the effect on aggression.
Yes. So she asked if we understand, I had indicated we understand the mechanism, or think we understand the mechanism by which the reduction in aggressive behaviors occurs. There was a study done at Sydney in Australia that showed that low GABA, GABA is a gamma-aminobutyric acid, it's a neurotransmitter, that if it's low use, it's associated with these aggressive behaviors in Prader-Willi. We think we amplify GABA signaling.